Welcome everyone. In the previous lecture, we were discussing about the early experiments related to the photosynthetic discoveries, right? The scientific background we are talking about. So, what I explained in the lecture number 2, I just wrote it on the board once again. We will just look into this and then we will get into the fresh discoveries, right? So, here in the lecture number 2, we discussed some experiments. The first experiment what we discussed is variegated leaf experiment, a croton leaf where green color patch will be there and other color patches also are there. Such a leaf is called variegated leaf experiment. Through variegated leaf experiment, we came to know that the green parts of the plant in the presence of the light, they will do photosynthesis, right? They covered half of that variegated leaf with a black paper. Their photosynthesis is not done, so they did not convert the starch, they did not convert the iodine color, whereas the remaining half of the leaf where the green patches are there, they are exposed, they have done photosynthesis, they made the starch, and that starch when reacted with iodine, it has given blue colored patches. That was the first experiment which tells that the green colored part of the plant in the presence of the sunlight will do photosynthesis. Experiment number 2, Moll's half leaf experiment. In this experiment, half of the leaf is kept inside a bottle where carbon dioxide is being absorbed. Light is there, green color is there, but there is no CO2. Since it is kept inside a beaker where carbon dioxide gas has been removed off by the KOH dipped cotton solution. Remaining half of the leaf is exposed to light, carbon dioxide is also there, green color is also there. The half of the leaf which is exposed is doing photosynthesis, it will prepare starch, that starch reacts with iodine solution, it changes the color. Whereas the remaining half of the leaf, the tip of the leaf which is inside the beaker where carbon dioxide is not available, there it did not do photosynthesis, then carbon dioxide is not available, it did not do photosynthesis, starch is not made, it is not changing the iodine solution. So Moll's half leaf experiments tell that carbon dioxide is required for photosynthesis. Third experiment, bell jar experiment conducted by Joseph Priestley. Joseph Priestley's bell jar experiment tells that when he kept a closed bell jar, in that when he kept a mice, and then when he has taken a candle, after some time the candle will turn off and he has seen that the mice also suffocated and it died. He told that the air was being fouled, the air quality has been disturbed by this mice and the candle, he told. The same setup he did, this time he kept a mint plant. When he kept a mint plant, he saw that the mint plant in the presence of light, it is doing photosynthesis. It's taking the carbon dioxide what the burning candle is releasing and the mice is releasing. Taking that carbon dioxide, the plant, the green part of the plant, mint plant in the presence of sunlight is doing photosynthesis and it releases oxygen as a byproduct. Now that oxygen is taken by this animal also, oxygen is taken by the burning candle also. The setup is clear. So he told that he don't know it is carbon dioxide and oxygen, even though he discovered oxygen, he don't know that the plants are releasing oxygen, he don't know that the animals release carbon dioxide. He told that whatever the burning candle and the mice have consumed, it has been restored by the plant. That is his inference. So Joseph Priestley's bell jar experiment, he told that plant will restore to the air, plant will give those components to the air what the burning candle and the breathing mice have removed. The next experiment, the similar experiment, same setup experiment was continued by another scientist. His name is Jan Ingenhousz. Jan Ingenhousz, when he conducted experiment, this time what he has taken is, he has kept the same bell jar setup in absence of light. In dark he made one setup and in presence of light he made one setup. He saw that plant is there, but plant can do photosynthesis only when light is there. Plant can release oxygen only when light is there. So when it is kept in dark, plant is not able to survive the mice, or plant is not able to lit the candle. Mice died and the candle turned off even in the presence of the plant. Whereas if you keep the setup in the presence of light, plant can do photosynthesis, it will release oxygen, with that oxygen mice is alive and the burning candle is lit. Alright, so sunlight is essential for the plant to purify the air is his inference. He also did another experiment, aquatic plant, bright light, it releases oxygen bubbles into the atmosphere, and he told that plants during photosynthesis they release oxygen Priestley did not tell they are releasing oxygen. Janningen House told that plants in the presence of light, they release oxygen, right? Green parts of the plants will release oxygen. So this is what he told. Next experiment, Julius von Sachs. Now Sachs experiment, he tells that plant will manufacture carbohydrate. It will manufacture glucose. So plant will produce starch, plant will produce glucose, and if it has to store it, it stores it as a polysaccharide called starch. And where is glucose made? He told there's a special pigment called chlorophyll. Chlorophyll is present in a special body called chloroplast. Chloroplast is a place where photosynthesis will occur. That is a place where glucose is made. That is a place where glucose is stored also. This is a conclusion given by Julius von Sachs. He told plant, by doing photosynthesis, they'll prepare glucose. It will convert the glucose into starch. Where is the starch present? Starch is present in chloroplast. Chloroplast has a pigment called chlorophyll. There glucose is made and glucose is stored, right? Next one. Experiment number 6. T.W. Engelmann has conducted, or he made the first action spectrum. Now what action I am doing? I am delivering a lecture like that. So Engelmann has delivered the first action spectrum. What is the action of the plant? Photosynthesis. So he told when plants are absorbing sunlight. So sunlight means visible light. Visible light has a range of spectrum starting from violet to red. VIBGYOR. Violet, indigo, blue. Okay. Green. After that, yellow, orange and red. In this range, he wants to see that which light is very much efficiently absorbed by the plant. When it absorbs more light, it has more energy. When it has more energy, it will do more photosynthesis. So that is the action spectrum he has seen. So on x-axis, this is the action spectrum. On x-axis, he has taken the wavelength of the light. On y-axis, he is checking the rate of photosynthesis. On y-axis, he is checking the rate of photosynthesis. How is he checking the rate of photosynthesis? He is measuring the amount of glucose, or he is measuring the amount of oxygen evolved. In terms of oxygen evolution, he is checking the rate of photosynthesis. He saw that at violet light, at blue light, photosynthesis is not much. Whereas violet light, indigo light, photosynthesis is not much. Whereas at the blue light, he saw there is a rapid rise in photosynthesis. Green light also photosynthesis is less. Yellow light also photosynthesis is less. Orange also photosynthesis is less. He again saw that the rate of photosynthesis increased at red wavelength. So plants show more photosynthesis activity at two particular wavelengths. One is at blue wavelength, other one is a red wavelength. By plotting an action spectrum, he told that plants absorb more blue light and red light and they do photosynthesis there. Okay. Now that, what is it called? It's called first action spectrum, was discovered by or explained by Engelmann, right? So go through the lecture 2 if you want to the clear clarity of what is meant by action spectrum. Then in NCERT they told the action spectrum matches with the absorption spectrum. The action spectrum matches with the absorption spectrum. What is absorption spectrum? Absorbing the light. Who will absorb the light? Pigment will absorb the light. What is a pigment present in the plant? Chlorophyll is a pigment present in the plant. So how many types of chlorophylls are there in higher plants? Chlorophyll A is there, chlorophyll B is there. Pigment definition itself is a substance which can absorb light. The chlorophyll pigment absorbs light. So then when we plot how much light the pigment is absorbing, then that's called absorption spectrum, right? So then we need to talk about two pigments, chlorophyll A and chlorophyll B. So I used blue color in NCERT textbook also. Blue color they have used for chlorophyll A pigment. Green color they have used for chlorophyll B pigment. So when they have plotted the absorption spectrum, means chlorophyll A is absorbing which wavelength more, means same blue wavelength chlorophyll A and chlorophyll B is absorbing, and again at red wavelength they are showing peaks, right? So they are absorbing more wavelength of light at blue wavelength and red wavelength, and they are doing photosynthesis also more at blue wavelength and red wavelength. This is action spectrum, this is absorption spectrum. Here you are checking the action, means photosynthesis, by measuring the oxygen evolution. Here you are directly checking the amount of light absorbed. The amount of light absorbed. When we check the amount of light absorbed, that is absorption spectrum. When we check the amount of photosynthesis done, that is action spectrum. In NCERT, the author has given a statement like the action spectrum roughly matches with the absorption spectrum. It is matching at the peaks. Blue peak, red peak, blue peak, red peak, it is matching. Whereas the remaining area, it is not have that much matching. Here it went down drastically down, but here it did not go that much down. So we can tell roughly the action spectrum and absorption spectrum matches. So this is what we have done in the previous lecture. Now take a screenshot if you want. Then we will start with our today's session. We will continue with few more experiments related to photosynthesis. Right. Now when we have to talk about the remaining experiments, what are the key discoveries left out related to photosynthesis, right? And how do we know that the final equation of photosynthesis, the balanced equation of photosynthesis, that we'll go to cover now. Photosynthesis is one of the important chapters in plant physiology. In botany also you can tell. So where a student needs utmost clarity, since chemistry comes here, right? So you need to understand it thoroughly, children. Then only you can solve the questions on the chapter photosynthesis. Now, in the mid-19th century, the key features of plant photosynthesis were established. What are the raw materials and all? We came to know when? By the mid of the 19th century. Next point we can tell, in MCET it is there. So, by the mid of 19th century, the key features of plant photosynthesis were understood by the scientists. What are the things which are required for photosynthesis were understood in the mid of the 19th century. Were understood by the scientists in mid of the 19th century. They told that the plants use sunlight energy to make carbohydrate by taking carbon dioxide and water. What are they telling? Plants will use sunlight energy to make the carbohydrate by absorbing carbon dioxide and water. So they told that plants, yes, they use, what do they use? Light energy. They use light energy. Why are they using light energy? To make carbohydrate, glucose, right? They make carbohydrate from carbon dioxide and water. So they were able to tell the equation, a rough equation, which we call it as an empirical equation, has been established, right? So which explains the total process of photosynthesis. So thus the empirical equation of total photosynthesis process can be written as, how can we write the equation? We can write the rough equation as carbon dioxide plus water. They told in the presence of light energy, it's going to make CH2O, carbohydrate, and it's going to release oxygen. This equation is called as a empirical equation, not a completely balanced equation. It was understood by the scientists when? By the mid of the 19th century. So they were telling that plants will absorb light energy, plants will convert carbon dioxide into carbohydrate, and the water is oxidized to oxygen. Plants, they release oxygen. Right, children? Understood till here? Later, one microbiologist is there. His name is Carolus von Neil. He is a microbiologist. Now he made a milestone discovery in photosynthesis that they will ask you. If they are asking you what is a milestone discovery in photosynthesis means, it is the experiment related to Carolus von Neil, who is a microbiologist. Let us write down. So we can tell a milestone discovery. A milestone discovery was the discovery done by a microbiologist. What is his name? His name is Cornelius von Neil. Cornelius von Neil. So what he has checked is there are some bacteria which are called as purple sulfur bacteria, which are called as purple green bacteria. These bacteria also will do photosynthesis like plants, it seems. He is a microbiologist. He'll work on microorganisms. Similarly, he worked on bacteria. Which type of bacteria? Purple sulfur bacteria, green sulfur bacteria. He checked that the purple sulfur bacteria are doing photosynthesis, and he tells that photosynthesis is a light dependent process. Yes, light energy is required. Even for bacteria also, he tells that light is required. So where the bacteria will convert carbon dioxide to carbohydrate. He tells that, let us write down. So in purple green bacteria also light energy is required. In bacteria also light energy is required for conversion of carbon dioxide to carbohydrate. For conversion of carbon dioxide to carbohydrate. Now check carefully here. Carbon dioxide is getting converted to carbohydrate. Hydrogen is coming extra. From where is the hydrogen coming? So he says that this hydrogen can come from any oxidizing compound. He tells that the hydrogen is donated by any oxidizing compound. It can be by any oxidizing compound. Since they are purple sulfur bacteria, green sulfur bacteria, they use H2S as donors of hydrogen. He tells that in purple sulfur bacteria, hydrogen comes via H2S, whereas in plant, water. In green plants, he tells that in green plants, who will donate hydrogens? Water will donate hydrogens. In green plants, water is donor. Since water is donor in green plants, we'll give the equation as carbon dioxide plus water gives rise to carbohydrate C6H12O6 plus O2. Hydrogen is going here and oxygen is being released out. Whereas this happens in green plants. Whereas in purple sulfur bacteria, he tells that water is not the donor, H2S is the donor. So then if we write H2S plus carbon dioxide, then what happens? As usual, hydrogen will go here, and instead of getting oxygen, sulfur will be coming out, right? So you will get glucose plus sulfur. We'll get glucose plus sulfur. So means he told that photosynthesis can be done without the release of oxygen. He told that photosynthesis can be done without the release of oxygen, and he tells that the oxygen which is evolved in photosynthesis comes from water, not from carbon dioxide. So what is his inference? What is his inference? He tells that oxygen evolved in photosynthesis. It comes from, the oxygen is coming from water, but not from carbon dioxide. Here carbon dioxide is there. Even though carbon dioxide is there, oxygen is not released, right? Only when water is there, oxygen is released. So this is his inference. Who is he? Is a microbiologist. He is a German microbiologist. He has done a milestone contribution, milestone discovery in the process of photosynthesis. He tells that without water molecule also photosynthesis can be done. Without releasing oxygen also photosynthesis can be done. What you need is just an oxidizing compound. So he has given an equation like 2HA plus carbon dioxide, 2HA plus carbon dioxide gives rise to C6H12O6 plus A. It comes out as A or A2, and water molecule will be released out, plus 2 water molecule. So this is the equation what he has given. We need to be clear with the equations also. In 2020 NEET, they have taken the nitrogenase action and they have asked what are the byproducts. Ammonia, hydrogen are the byproducts. So children think that only ammonia is a byproduct, but with ammonia, hydrogen is also released. The first option is only ammonia na, or ammonia with hydrogen also na. So like that they can ask us, in von Neil experiment, who is a hydrogen donor? It can be any oxidizing compound like H2S or H2A. The equation is 2H2A plus carbon dioxide if you take, you will get 2 A's, you'll get 2 water molecules and you'll get a carbohydrate, CH2O. You can go, right? So take a screenshot, we will continue. Right? Later, using radioisotopes techniques, Ruben and Kaman, they proved that the oxygen which comes in photosynthesis comes from water only. He just told it, right? But the proof came through radioactive experiments done by 2 more scientists. Their names are Ruben and Kaman. So we can tell that in 1941, in 1941, it's not rumen, it's a part of stomach. Ruben and Kaman. In 1941, Ruben and Kaman have conducted radioisotope technique. They have used radioisotopes to prove that oxygen during photosynthesis, it comes from water only. What did they do? They have taken carbon dioxide molecule, they have taken water molecule also. The oxygen which is there in the water molecule is a radioactive oxygen. It is having radioactive O18 isotope. So this is normal oxygen, this is radioactive oxygen. So when they have taken it, so they got glucose C6H12O6, then you will get 6 O2 molecules and then we will get 6 water molecules, 6 CO2 will be there and 12 H2O will be there. This is a complete balanced equation of photosynthesis. 6 carbon dioxide molecules are reduced by 12 water molecules to give 1 glucose, 6 oxygen and 6 water molecules. Now when they are checking where is this O18, here in carbohydrate the oxygen is not O18, means in carbohydrate the oxygen is coming from carbon dioxide only, whereas in O2 the oxygen is O18. So this is enough to tell that the oxygen which is evolved in photosynthesis comes from water. So the O18 isotope they have observed with oxygen, whereas this is reactant water, that is product water, right? So we can write it as substrate water. This is substrate water and this is called product water. Now, in product water, hydrogens obviously will come from here only. The oxygens, H2O, the oxygens are not O18. So that means that oxygens which are in product water are not from water, they are from carbon dioxide. So in product water, this oxygen is from carbon dioxide again. It is this oxygen. CO2 oxygen only is coming out in water molecule, whereas the oxygen which is coming out here as a gas which we are breathing comes from water molecule. So this is radioisotope experiment to prove that. What's the inference? Come on, tell me, I'll write down. What is the inference? The inference is the oxygen evolved during photosynthesis comes from, the oxygen evolved comes from water but not from CO2. It is proved by using radioisotopes experiment. Understood, children? Now how do you came to know that carbon dioxide is required for photosynthesis? Moles half-leaf experiment, right? So carbon dioxide is required for photosynthesis. How do you came to know? Moles half-leaf experiment. How do you came to know? With which experiments you came to know that water is required for photosynthesis? De Saucer. So in 1804, another scientist, his name is, right, De Saucer. His name is De Saucer. He told that water is required for photosynthesis? Who told carbon dioxide is required for photosynthesis? Moles, Van Mol, through one Mol's half-leaf experiment. Who told that water is required for photosynthesis? D. Saucer told that water is required for photosynthesis. Now, who told that plants have a pigment called chlorophyll, and chlorophyll will absorb light energy? So there's a scientist called Pellitter. There's a scientist called Pellitter and Caventou. So they observed chlorophyll as a pigment present in plant and they named it also as chlorophyll. So we can tell in 1818 Pellitter and Caventou. Pellitter and Caventou discovered the plant pigment chlorophyll and named it also as chlorophyll. They discovered plant pigment and they named it as chlorophyll. They named it as chlorophyll, right? Understood till here? Now, what did Blackman do? There's one more scientist. His name is Blackman. Now, what did he tell? Like, he tells that like light is required for photosynthesis, but then he tells like dark is also required for photosynthesis. He tells that in photosynthesis there is a dark phase also. Blackman observed dark phase. That means in photosynthesis light phase is there, dark phase is there. Blackman observed dark phase in photosynthesis. Blackman observed dark phase in photosynthesis. He told that the photosynthesis is biphasic. Light phase will be there, dark phase will be there. And he also proposed law of limiting factors. He proposed law of limiting factors. Means if the carbon dioxide is limiting, photosynthesis will not happen. If the water is limiting, photosynthesis will not happen. If the light is limiting, photosynthesis will not happen. So this is called law of limiting factors. Who proposed law of limiting factors? Blackman proposed law of limiting factors. Now what did he also tell? He tells that in photosynthesis there is a dark phase also. Who tells that water is required for photosynthesis? D. Saucer told water is required for photosynthesis. Who told that carbon dioxide is required for photosynthesis? Von Mol. Through Mol's half-leaf experiment, he tells that carbon dioxide is required for photosynthesis. Who proved that the oxygen which comes after photosynthesis comes from water? Ruben and Kaman through radioisotopes technique. Take a screenshot. We will continue. Right. There's another scientist, his name is von Meyer. He told that plants will convert sunlight energy into chemical energy and that energy will be used to convert carbon dioxide into carbohydrate. So another scientist is von Meyer. Von Meyer, he tells that plants absorb sunlight energy. Plants absorb sunlight energy and they convert the sunlight energy into chemical energy. And they use this chemical energy to convert carbon dioxide into carbohydrate. This is what is a conclusion given by von Meyer. He tells that plants absorb sunlight energy, which is a physical energy. They convert that physical energy into chemical energy. They use that chemical energy to make glucose. Understood till here? Then we can tell that Emerson and Arnold performed flashing experiments in presence of light, in absence of light, in presence of light, in absence of light. This is called flashing experiment. Alternatively, light and dark. So they came to know that plant equally requires light also, dark also for doing photosynthesis, and then they told photosynthesis is biphasic. Okay, so we can tell in 1932 Emerson and Arnold. In 1932, Emerson and Arnold, they have conducted intermittent flash experiments. They have conducted intermittent flash experiments. They are exposing the plant to light, they're taking it to dark. Again exposing to light, taking it to dark. Alternatively, light, dark, and they came to know that plants require this for doing photosynthesis. Then these people told that photosynthesis is biphasic. Means they are telling in photosynthesis there is a phase which is called as light phase, and they're telling there's a phase which is called dark phase. You remember Blackman also told the same thing, that there is dark phase in photosynthesis. These people proved it. Who proved it? Emerson and Arnold. They proved that like photosynthesis requires a light phase also, dark phase also. Right children? Understood till here? Then another scientist, his name is Warburg. His name is Warburg. He tells that in presence of oxygen, photosynthetic rate decreases. So for photosynthesis, carbon dioxide is required, no? If carbon dioxide is less and oxygen is more, plant cannot do photosynthesis, it seems. Plant cannot do photosynthesis. That means we are doing good to the plant. We are releasing carbon dioxide and we are giving that carbon dioxide to the plant so that it can do photosynthesis and it can make energy. So Warburg effect. What is meant by Warburg effect? Warburg effect. He tells that in presence of oxygen, in presence of high levels of oxygen, in presence of more levels of oxygen, plant is unable to do photosynthesis. Plant will not do photosynthesis. Instead, it will do photorespiration, it seems. Plant will not do photosynthesis, but will do photorespiration. Instead, it will do photorespiration, which is called as Warburg effect. Understood? Understood till here? Then there is another scientist. His name is Melvin Calvin. Melvin Calvin. So Calvin, what did he propose? He proposed C3 cycle. He told that when carbon dioxide is converted to glucose, which is a 6-carbon compound, directly from one carbon compound you cannot make a 6-carbon compound. He tells that the first stable compound is a 3-carbon acid. When carbon dioxide is reduced to carbohydrate, from one carbon we cannot directly get a 6-carbon compound. It goes via a 3-carbon acid. So that is why it's called C3 cycle. Who proposed the C3 cycle in photosynthesis? Slowly when we go in the next classes you will understand what is C3 cycle. Calvin told that plants prepare glucose by C3 cycle. Then another scientists are there, Hatch and Slack. They told that there are some plants where when carbon dioxide is converted to carbohydrate, they don't make C3 acid but they make C4 acid. They don't make C3 acid. That means there are some plants, instead of making C3 acid, they can make an acid with 4 carbons. It's called C4 acid. Then those plants are called C4 plants. All these plants which can make C3 acid are called C3 plants. Hatch-Slack pathway means it is C4 pathway. Hatch and Slack observed that in some plants like sugarcane, amaranthus, those plants when they are converting carbon dioxide to carbohydrate, they don't make the glucose directly, they make an intermediate called C4 acid. Understood children? Then there's another scientist, I'll write down here, Arnon. There's another scientist called Arnon. He explained photophosphorylation. He explained photophosphorylation. What is photophosphorylation? Phosphorylation means ATP synthesis. Photo means using sunlight to make ATP. Using sunlight to make ATP is called photophosphorylation. Arnon explained the process of ATP synthesis in photosynthesis. It is called photophosphorylation. I think we covered all the details, historical details related to photosynthesis. So in the next lecture, we will talk about chlorophyll and its role in photosynthesis. We discussed Moll's half-leaf experiment. We discussed variegated leaf experiment. Bell-Jar experiment, Jahningen-House experiment, Sachs experiment we completed, Engelmann experiment we completed, then we Van Niel experiment we completed. After Van Niel experiment, Ruben and Kaman experiment we completed. De Saucer, he told that water is required for photosynthesis. Pellitter, they discovered chlorophyll, they named it as chlorophyll, that we understood. Von Mayer, what did Von Mayer told? He tells that sunlight is converted to chemical energy. Warburg effect, what is it called? Photorespiration. Calvin, C3 cycle. Hatzlack, C4 cycle. Arnon, he explained synthesis of ATP in photosynthesis. So if you like the content, like, share and subscribe to my channel. Thank you.